Hi, I'm Dane Boucher.
Hello, I'm Momal Ijaz.
We also have a third member, Sheng Yang, but he's not here to present with us today. So we're doing a paper called AlphaFold by Google DeepMind. So to give you some background, proteins are these chains of small molecules, amino acids. You take these small amino acids and chain them together, and you get a long molecule, a protein. So proteins are essentially the building blocks of life. Your body is full of them, and nearly every function of the body is dependent upon a certain protein. So what's special about these long proteins is they fold up to form unique shapes. So in fact, misfolded proteins can actually are thought to be the cause of certain birth defects related to viruses, like the coronavirus. So understanding how a protein structure actually folds and takes shape is very, very important. Theoretical work has attempted to actually predict these protein structures just given an amino acid sequence. So on the left there, you can see a picture of a protein formed by chains of amino acids. So these in turn form certain shapes at certain parts of the protein, such as alpha helixes or sheets. So these shapes of the proteins are then used, once again, just for all sorts of functions, and the shape of the protein dictates exactly what that protein does, such as maybe like interacting and signaling and transcribing DNA. So CASP is actually a competition to predict protein structure based solely on these protein sequences. It actually has two categories. One is template-based modeling, which we actually have a similar protein with a known structure, and free modeling, in which we don't have actually any proteins whatsoever, similar proteins whatsoever. So what's interesting is AlphaFold is actually an exclusive free modeling method, and is actually it was entered into both competitions, even the template based, even though it was a totally free model. So as you can see at the bottom there, it won by a very large margin compared to all the other competitors.
Okay, so let me walk you guys through a bit about the problem definition. So the high-level idea that we are trying to implement is that we are going to be given a protein sequence and we have to predict it using our model that how it is going to fold up and what 3D shape is going to take. So, the motivation for choosing this topic was that nature has encoded the amino acid sequence information of a protein into the genes of a living organism, but the information on how these proteins are going to fold up is not there. And this is a really important piece of information because that helps us determine what the function is there that the protein is going to perform. And by knowing the function, scientists can actually discover and develop new drugs for all sorts of diseases. That also includes COVID-19, SARS-CoV-2, and other virus-based diseases for which there ultimately is no drug released or discovered up till this day. The problem definition is that we are going to have a protein sequence, a chain of amino acids, and some features that are going to be extracted or engineered using some biochemical and machine learning based techniques. We are going to give this as an input to our deep learning model. The output of the model is going to be a predicted 3D protein structure or shape in form of 3D points of the amino acid. The intermediate output of the network is going to be a distogram or distance histogram between the possible pairs of the amino acids in the sequence, and also the torsion angles between those pairs, which ultimately will help us make the final structure.
So moving into the methods, we start by taking these features and process them and do feature engineering on them. Once we have all our features, we feed them into a convolutional neural network. This then predicts a bunch of factors, the most important being distance. Finally, once we have the distance, we can do structure realization, such as taking the structure and sort of relaxing it using gradient descent to actually get the final 3D shape. So from a high level view, we take our protein sequence composed of a bunch of amino acids. We then compare that using a database to known proteins with known structures to then build various features. We then take all these features, feed them into the neural network. This then predicts the distance and the angles. And we actually use the angles to sort of initiate the 3D structure, and then the distance to actually minimize the potential. So we then use gradient descent to then minimize the potential and actually get our 3D shape. This final stage, I like to think of it like you have a spring that's sort of stretched out, and then we're just sort of relaxing that spring to minimize the potential. So to get a little bit more in depth, we have our sequence. We then have our sequence database. We use the two to do something called multiple sequence alignment, which means finding similar sequences and comparing them and looking for how they differ. H8 splits and psi blasts are used to actually get us the multiple sequence alignments features. So these are programs that were developed before this paper and actually exist out there, and they were used throughout this process to give us those additional inputs and features. So we take all these, feed them into a deep residual network. This is the convolutional network. And we get our torsion, that is angles, predictions, and our distance predictions. Once we have the two, once again we use the torsion to initiate the model, and then both actually do the potential optimization using gradient descent. And then we finally have our 3D shape of the protein given to us by the positions of the carbon atoms of the amino acids. So this is another visualization. So we have our features, which in this case is a 2D matrix of features. So the coordinates are actually the comparisons between two different amino acids of the protein. And the diagonal is actually the amino acid compared to itself. So taking all that data as a 2D structure, we feed that into 220 residual convolutional blocks. That's just our neural network full of convolutions. And then our output is the distogram, which is the prediction of distances between the amino acids. So, of course, that diagonal there is the distance from the amino acid to itself, which is 0. So that's why it's always a solid color. And the other ones are the distances between the other amino acids. And then lastly, we take that distogram to then apply our gradient descent process.
Okay, so let me quickly talk about the distance prediction output of the convolutional neural network Dane just talked about. So the deep neural network architecture that we are using is a two-dimensional dilated convolutional residual network with 220 residual blocks. So it's a deep network that has already been trained by the DeepMind, and the weights are shared. So this is a dilated CNN. Dilated means we can skip the rows in the input to get a more broader range of input to cover. And residual network is something which is a very famous architecture, and the blocks of residual networks are being used um, in the deep ResNet, in the deep architecture here. So here's a zoomed in view of a single residual block that is used in the network. So we get the input, which is first passed through a batch normalization layer and then passed through an activation that is exponential linear unit. We scale it down to 64 dimension, the same sequence again. Then we pass it through a 3x3 dilated convolutional layer, which is then passed through again the same sequence, some more non-linearity, and we project it up to the same dimension, that is 128, same as input. The part that we need to focus on here is this skip connection, which actually lets the network, these deep networks learn actually really well and avoid the vanishing gradient problem. Here is how the output of the network is going to look like, and this is a figure we have taken from the paper. So the above three images are the distogram or the distance histogram images for the ground truth of these three different proteins, and these three are the predicted ones by the AlphaFold's model. So the distance unit here is angstrom, which is the bond length, or probably the distance between the atoms. And the lower or the lighter colors are referring to the lower values of the distance, which means if lighter is the color, then most probably there is going to be a bond there. So this is the kind of output that we want our network to predict for us. And this, including with some other torsion angle input, is passed into into the potential function, which gives us the relaxed or the stable protein 3D structure. So we can see it here in these three figures, the structures that are predicted for these proteins. The green ones are the ground truth and the blue ones are the predicted ones, and they're quite close actually. And this is a beautiful simulation or animation of how the gradient descent method actually helps this protein chain get a stable and a relaxed position by descending over the potential function that's been calculated for this. And this is for specifically T108 protein. So thank you so much for your time.